Hi everyone, welcome to part 2 of our lecture 2 on our course on plant specialized metabolism. So in this video I want to follow up on the first part of the lecture where we looked at terpenoids as our first large class of specialized metabolites, and we looked more generally at their structural and functional diversity as well as at the sort of general principles of how these compounds are biosynthesized with a clear focus on plants and how this is achieved in plants. What I want to do in the second part is to actually look at some of these subgroups of terpenoids, more specifically to understand the commonalities and differences in, again, the structure and function and the biosynthesis of this really large group of natural products. So let's start with the monoterpenoids as one of the smaller, more volatile group of terpenoids. They are a very large subgroup. They have 10 carbons, that's how they are defined. So the subgroups of terpenoids in general are defined based on the carbon numbers in the hydrocarbon tails. So in this case, 10 carbons are called monoterpenoids. And monoterpenoids actually are formed almost completely in the plastids. Right? We had talked in the first part of the lecture about the central intermediates of all terpenoids, these 5 carbon isomeric precursors, IPP and DMAPP, originating either from the plastidyl MAP pathway or the cytosolic mevalonate pathway. In case of the monoterpenoids, they are derived almost exclusively from the plastidyl MAP pathway, through the head-to-tail condensation of 1 unit IPP and 1 unit of DMAPP, you're generating this linear 10-carbon prenyl diphosphate substrate, general diphosphate or general pyrophosphate. This is the central substrate to all of the monoterpenoids. And so this is occurring in the plastid for almost all of the monoterpenoids, with the exception of those that are further functionally modified by P450s, which would then be occurring at the ER. But for the most part, for the reactions we're seeing here is happening in the plastids. The enzymes that are now involved in converting this linear precursor and defining the initial structural scaffolds of the monoterpenoids are, again, our terpene syntases. And in case of the monoterpenoids, these are formed almost exclusively, again, by these monofunctional class 1 enzymes. And so these are actually a specific kind that have lost in the course of evolution this additional gamma domain here. So these are 2-domain enzymes where we have the active site sitting here in the C-terminal domain. This is your class 1 active site. So for the most part, all the reactions that we will see and all the different scaffolds are generated by these type of 2-domain class 1 terpene syntases. Structurally and functionally, these enzymes are very similar. We had talked about them being structurally very similar. And while we have a large diversity of structural scaffolds in the group of monoterpenoids, the general mechanism of the enzymes is the same. What differs is the specific cyclization and rearrangement reactions that we're seeing. So I have a video here that was actually prepared by another group showing how the reaction would work in this case for the production of limonene, which is sort of a major monoterpenoid that you will know from especially citrus fruit. So the flavor and the fragrance of citrus fruit is limonene for the largest part. And so this illustrates quite nicely how the biosynthesis is actually accomplished through the terpene syntases. And you start out right with your general diphosphate linear intermediate, right? The diphosphate group is sitting right here. This is your 10-carbon hydrocarbon tail. Now assume this molecule is entering the active site. It starts to actually be folded in the active site. Then we are cleaving off the diphosphate group. We're generating a carbocation here that will allow us to delocalize the electrons between these 3 carbon atoms here. And basically the rest of the reaction now is to rearrange the molecule and eventually to terminalize the carbocation and stabilize it. What is happening in this particular reaction is that actually in the active site, the cleaved diphosphate group acts as a counterion. In some cases, it will actually bind, like in limonene. Not always it does. The exact function is actually not clear. And then you have a number of rotations and movement of the carbocation to facilitate this full series of rearrangements. In this case, we're actually facilitating cyclization, removing the carbocation over, and then you can neutralize the carbocation by deprotonation at the end. This is the most common form of neutralizing the carbocations in terpene synthesis. There are few examples where it's actually being achieved by water quenching, so bringing in a hydroxy group through a nucleophilic attack here. But this is essentially the mechanism by which you can make these compounds, in this case limonene. So it's a really nice illustration, but in general, it works through binding the substrate, cleaving off the diphosphate group that will act as a counterion to the generated carbocation, and then rearrangement of the carbocation through cyclization and or various rearrangements within the molecule to generate all the different scaffolds. And this is sort of what is illustrated here now. Here you have, again, your linear precursor, the general diphosphate. You're cleaving off the diphosphate group, you generate this first carbocation here, the general cation. From this, you already can make a whole bunch of acyclic intermediates. You can actually now bring in back the diphosphate group as was shown. That can be removed again. You're generating a secondary carbocation and another one here. So it's just a series of moving the position of the positive charge around in the molecule. And from here, through simple rearrangement and final termination of the carbocation, you can generate this entire series of different terpene scaffolds. And so these can be all different terpene syntheses. So all they have is slightly different contours of the active site. They use the same mechanism to achieve all of the structure diversity. And you can see here that, right, like limonene, as I mentioned, is very abundant in citrus fruit. If you actually generate something like terpenine here, so a slightly different rearrangement, or you might add another hydroxy group here. So here you would quench the carbocation with water, not through deprotonation. You get, for example, terpineol. This is a major terpene in the rose fragrance, for example. Down here, pinene, they are very common in your conifer resins. And so they are very strong antimicrobials. There's actually some really interesting work also on using dimerization of pinene to generate jet fuels. So I'm just sort of trying to highlight a few sort of major functions and applications here. But the bottom line is you can achieve enormous structural diversity simply by coordinating the rearrangement of these intermediary carbocations in a slightly different way, but tapping into the exact same mechanism, essentially. And again, like in terms of applications, right, naturally you would find this, for example, limonene in citrus fruit, but you will also find limonene in many of your cosmetics, your dish soaps, and so forth. Obviously, many of these terpenes that have nice scents, those you will find in all of the higher level perfumes. Right? So there are many applications of these. When it comes to their physiological functions, they're obviously not for perfume, but all of these monoterpenoids are very easily evaporate. They're volatile compounds, and so they are often part in the more long distance type of plant environment signaling. Many of these are involved in pollinator attraction, especially terpineol, others. Some also are antimicrobial, such as pinene, I mentioned that. And then again, others can be involved in tritrophic and other plant-plant interactions. Linalool is one example, right, where a plant, if it's attacked, can actually use these long-distance volatile compounds as signals to attract predators to whatever caterpillar, for example, is attacking the plant. And there are also some that they are volatile that are released into the rhizosphere. There are some examples that they accumulate and seem to have an impact on the assembly of the microbiome. That's still very new research. It's not exactly understood, but there's increasing support that there are additional roles of these compounds also below ground. For the above ground long distance signals, the exact mechanisms on how this works are actually not understood very well. What we do know in most cases is that they will interact with these odorant binding proteins in an insect, for example. So these odorant binding proteins will sit in the antenna of an insect, for example. The volatile will come in, will be recognized, and will be bound by these small proteins. They're similar to lipid transfer proteins. They bind hydrophobic compounds. And so once bound here to the molecule, these odor proteins can be recognized and can bring the metabolite toward different kinds of receptors, can release them. that would be the recognition set. How this exactly works, again, is outside of the scope of our class. I'm just sort of trying to highlight here in general how these molecules will be part of the interaction system with, for example, insects. Just to give one example on their antimicrobial and defensive capacities, again, I'm using the example here of the conifer bark beetle interaction and the huge issues that we have across Canada and large parts of the US by now, as well as Europe. And so here, the resin that is really rich in diterpenoids, and we'll get to those in a bit, but also monoterpenoids are really toxic to the insects, as well as some of the fungal pathogens that come along with the insects. And so we really have to look here at sort of a triangular relationship where these resins have evolved to defend the plant against the stem-boring bark beetles. Again, the polymerization part of this resin, this would be the higher molecular weight diterpenoids. The toxic part to a large degree are the monoterpenoids, especially pinene is toxic to these beetles by a mechanism that, again, we don't really know to a large degree how that works. But they also interact with a fungus here that actually is entering the tree through the holes that the beetles drill into the stem. So the beetle here acts as a vector for the fungus. And so this blue stain fungus is very common, and it's actually often what kills a tree. It's not as much the beetle, it is often more the fungus that is associated with the beetle that actually kills a tree. And so there has been a lot of research, if you're interested in this topic, coming out of Canada and the US, a lot of genomics and biochemical work that is trying to understand this triangular relationship between the tree, the beetle, and the fungus. And one result that I really want to highlight here actually is some of the capacities of the fungus to deal with these defenses. And so what has been done is actually that they sequenced the genome of the tree, the beetle, as well as the fungus. They did a lot of co-expression and biochemical studies. And what they found is actually that this fungus can grow on terpenoids as a carbon resource alone. So it's not dependent on sugar. It can actually eat up these defenses as carbon resources. And you see this here on the right as the growth rate of this fungus here in presence of the control only, where you would have simple sugars. And then if you grow them, for example, of pinene, carine, limonene, all of these are compounds that are abundant in conifer resin. The fungus is a little bit deficient in growth, but it can grow. And in addition to that, it actually, they used the genomics tools to identify that this fungus also has an ABC transporter that is capable of actually effluxing surplus monoterpenoids from the cell. So it's evolved a double defense against these defensive chemicals. Is really a fascinating example of co-evolution. And I'm happy to share the literature on this if people are interested. All right, so just sort of in a nutshell, I don't want to go too long here about some examples on the monoterpenoids. The next one up in terms of carbon size is the sesquiterpenoids. These are your 15 carbon terpenoids. And so while we have many commonalities in how they're made, there are some key differences. The first one being that sesquiterpenoids, along with the triterpenoids, are actually derived from the cytosolic mevalonic pathway. So these compounds actually don't ever touch the plastid. They are completely formed in the cytosol through initial condensation now of 3 of these 5 carbon units to generate farnesyl diphosphate as a central precursor to the sesquiterpenoids. The mechanism essentially is the same as I'd shown you for the monoterpenoid synthesis, the same type of class 1 terpene synthesis. Again, most of these, but not all, are these 2-domain type class 1 terpene synthesis. The principle also is the same. For the most part, you will be binding the substrate in the active site. You will cleave off the diphosphate group, generate different carbocations, and you have the possibility for a number of different carbocation rearrangements. The diversity is enormous in the sesquiterpenoids. So this is actually an example of all the products that one terpene synthase can generate. So what I want to highlight here actually is that they're not always super precise and are one substrate, one product enzymes. Many, especially mono and sesquiterpene synthetases, are multi-product enzymes. So their active sites are essentially a little bit more relaxed, and they allow for a large diversity of products to be formed. This, I guess, it's not quite clear why this is a selective advantage, but you could easily imagine that it allows you to increase chemical diversity and also have catalytic efficiency in being able to produce an entire cocktail of defensive compounds using simply one enzyme. And again, just to highlight a few functions here, generally their physiological roles overlap largely with the monoterpenoids. They're similarly volatile, used both locally but largely also in long distance signaling and defenses. And some examples here are caryophyllene. That's a very prominent sesquiterpenoid you find, for example, in tomato, in maize, many other crops actually. And so caryophyllene actually functions both as an outright defense compound that deters certain insect pests, but it also is involved in tritrophic interactions where plants actually emit it and attract parasitic wasps. And it's emitted upon the chewing of certain herbivores on the plants. There are different kinds. So upon the caterpillar here, the herbivore chewing on the leaves, caryophyllene is released into the atmosphere, travels fairly long distance, and it can attract these parasitic webs that are predators to the herbivore. So there's is one of the common functions of caryophyllene, and is really well understood. There are others then, like germicrin D and many of these others that we find, for example, in sunflower. These are precursors to more complex terpenoids that form lactones and have strong either anti-herbivore or antimicrobial activities there. Then some of those that are not much further modified, like bisabolene here, this is a typical hydrocarbon. This is a really prominent example of a sesquiterpene that is used as a biofuel precursor, for example. But there are many of them. It's a large group, so I'm just highlighting a few examples here. Then moving on to the diterpenoids, now things change a little bit. We have, again, 5 more carbons. We're moving actually back into the plastid. So the scaffold formation of the diterpenoids is derived largely from the MAP pathway, and it's occurring in the plastid. Again, we said earlier in the lecture that most of these compounds are functionally modified, largely by P450s. So diterpenoids are a classic example where we have the scaffold formation occurring in the plastid and the functional modifications occurring at the membrane, at the ER membrane, the surface thereof on the cytosolic side for the most part. What is different now with the diterpenes in many cases is that only a few diterpenes are actually formed by monofunctional class 1 enzymes. It's getting much more complicated here. We have a few examples like the taxanes, which are your precursors to taxol. Those are produced outright by class 1 type terpene synthesis. But we also have, especially the more ancestral ones that we find in the conifers here, we actually find these by functional diterpene synthesis that have 2 active sites, a class 2 active site and a class 1 active site. They're generating similar kinds of compounds, but we're using bifunctional enzymes here. And we only find those in the evolutionary older species, such as gymnosperms, bryophytes, lycopods, fungi, for example. In the higher plants, now in the angiosperms, what we're seeing is actually that we're using monofunctional enzymes. And what we're actually using is pairs of monofunctional enzymes, one being a class 2 diterpene synthase that has the active site in the N-terminal domain, and one a monofunctional class 1 diterpene synthase that has it in the C-terminal domain and catalyzes a reaction much like we had seen before for the mono and sesquiterpene synthases. Now here we're using pairs, and so now the first one, the class 2 diterpene synthase, will catalyze the first reaction, which works somewhat differently. It's actually protonation initiated. It does not cleave the diphosphate group. What it does is that it actually uses protonation to generate intermediary carbocations, and then uses rearrangement to generate a bicyclic intermediate. And this intermediate then will go to the class 1 diterpene synthase. This will undergo now the classic reaction mechanism that I showed you for the mono and sesquiterpenoid synthases. cleavage of the diphosphate group, generating a carbocation, and then a whole slot of different possible rearrangements. So we're just using a pair of enzymes here. If I go back here, what that helps you to do is, right, that you can use two enzymes, the first one generating a whole lot now of different intermediates, and these can be taken up again by a group of different class 1 enzymes. And so if you imagine now that most plants have families that are multi-gene class 2 and class 1 diterpene synthetases, these can actually interact in different combinations. So you can have different class 1 enzymes interact with different class 2 enzymes. And so this allows you to be much more dynamic in which enzyme do you have interact with which other, and it allows you to really be essentially modular. You can generate modular pathways using combinations of functionally distinct enzymes, and without having to produce a new enzyme, evolve a new enzyme, you can generate a much larger range of different possible scaffolds of diterpenes. So the advantage really is flexibility and modularity to enhance product diversity simply through combinatorial biochemistry. There's a large group of these, more than 12 to 15,000 of these compounds are known in plants. Their functions are differing now from the mono and sesquiterpenes, that these are typically higher molecular weight and are much less volatile than the smaller terpenoids. And so the large part of the functionality of diterpenoids is defensive, with the exception of our gibberellin phytohormones, right? These are operating as hormones in general metabolism. All others are specialized. The large part here is they can act as anti-feeding compounds. There are well-published examples, for example, in maize, where diterpenoids function against the European corn borer. Many of those have antimicrobial activity against bacteria or fungal pathogens. There are many examples of that in the literature. There are examples in rice, for example, where root-specific diterpenoids have allelopathic functions, so they're suppressing actually the growth of competing plants around rice. Again, there are some recent examples in the field that also show that diterpenoids that are in the rhizosphere have impact on the composition of the microbiome. And there are very few examples, one specifically in Arabidopsis, dihydroabiatane, the aldehyde, seems to be involved in SAR, in systemic acquired resistance, as a long-distance signal. There's still ongoing research on how that actually works, but it's one of the examples. And there are probably more out there that diterpenoids also have signaling functions rather than outright toxic functions. And again, the mechanisms by which diterpenoids act are largely unknown. There are many groups, including my own, that are working on this, but there's not much knowledge on the precise modes of actions of these compounds. Much sort of building also on their really diverse physiological functions, diterpenoids also have a huge range of applications. I'm highlighting just a few here. Many diterpenoids, such as Taxol or forskolin here, are FDA-approved drugs, and there are many more in the pipeline. Taxol being a chemotherapeutic drug, forskolin is a C-AMP regulator for use in treating cardiovascular diseases. Salvinorin A is an interesting example that is undergoing clinical trials, for example, to be used as for the treatment of drug addictions. It actually turns out that salvinorin A acts on the same receptor as morphine does, but it does not have the same addictive properties as morphine. So it's of great interest at the moment for treating drug addictions and other neuropsychological disorders. But then also outside of the pharmaceuticals, diterpenoids are used in industry in almost all realms. Stevioside, you probably know, as a natural sweetener. Carnosic acid actually used industrially as a food preservative. Sclareol is a diterpene alcohol that is used heavily as a base note in the perfume industry, and so on. These are just a few examples of the many applications that we have for diterpenoids. Then moving on, we'll keep adding carbons here. So we're adding 10 this time. There are actually 25 carbon terpenoids. I'm not going to touch on them here, but they do exist. More commonly, though, is that you're jumping to the 30 carbons now, the triterpenoids. And so what we're doing here, the upstream pathway is a little bit different in that we don't bring in additional units of IPP. What we're doing to make triterpenoids actually is that we're doing a condensation of two 15-carbon FPP precursors instead. So we're condensating the two C15s, making one acyclic C30 precursor. And because sesquiterpenoid metabolism is occurring in the cytosol, we're using this precursor. Triterpenoid metabolism also is occurring in the cytosol and not in the plastids. So what we're doing here is we're doing the same kind of condensation of these units. What we're doing, though, is we don't do a head-to-tail. We're doing a head-to-head condensation. So it's just a slight different rearrangement, but the same principle that you're using the cleavage of the phosphoanhydrate group here to start the reaction, to release energy, to facilitate the condensation. You generate the positive charge here, it's a C1. And then it's a little bit different on where you are actually attaching. The head to tail, you would be attaching basically at the end of this molecule. Now this is a little bit longer here, right? This is your 15 carbon intermediate. You will actually undergo condensation at this carbon here. You're shuffling the positive charge around and you're doing a rearrangement that I will not ask you to know. The chemists in the group might have a specific interest here, but I will not ask you. And then you're simply doing another reduction reaction and you're ending up at this long hydrocarbon tail here. And that is squalene. That's your basic 330 linear hydrocarbon. That is a precursor to all of the triterpenoids, but is also, for example, in fungi, a key membrane component. And that again becomes important if you're thinking about metabolic engineering, right? If you're tapping into squalene formation for making triterpenoids, you have to deal with the need of yeast as a fungal species of needing squalene. So you actually have competing pathways there that you need to take into consideration for metabolic engineering. Then how do we make triterpenoids? Based on this initial scaffold, again, we have our tailoring reactions. And for all of the diversity of triterpenoids out there, there are basically two enzyme classes that do the job. The first one being a variety of different cytochrome P450s. Again, I will not ask you any of the details. There are many, many pathways out there. In general, you are doing a cyclization of squalene here. Depending on which enzyme is doing the cyclization, you can generate backbones that can go into the sterols. If you're going through an amyrin synthase here, you're actually generating a 5-ring molecule from the C30 precursor that is beta-amyrin. And this now is a central precursor to all of the specialized triterpenoids. This will undergo further oxygenation now by a number of different P450s. And if you look at some of these structures here, you will see that you have different kind of oxygenations, everything from hydroxylation to carboxylation and position specific. Okay. What is unusual about the triterpenoids in the realm of terpenoids is that they almost always are glycosylated. So a mono, sesqui, and diterpenoids, glycosylations can occur, but they are actually more rare, at least for the compounds that we know so far. Stebioside would be a good example for an exception here that is heavily glycosylated as a diterpenoid. But for the most part, what we know from the compounds as they occur in the plants, they are not glycosylated. For the triterpenoids, this is different. Almost all of these are glycosylated. So the next class of enzymes here that will act on these oxygenated intermediates are various kinds of glucosyl transferases that now will add one or more sugar moieties to these oxygen functions. And so with that, you end up with an enormous structural diversity of triterpenoids. What they do, largely triterpenoids, if you ever tasted them, they're bitter. So those among you who might like bitter lemon or things like this, or zucchini, sometimes you will actually find those in the plant. So if they're the bitters, these are triterpenoids. And so these essentially function as antiphedans. Many triterpenoids also have antipathogen, antimicrobial activities. And again, there's some research out there, very nice research recently from Anne Osborne's group, for example, that also provide evidence that triterpenoids that are specific to the root system, again, are functioning in the assembly of the microbiome. One example that I want to show here is actually work in cucumber, where people have used genome sequencing combined with QTL studies and biochemical studies and enzyme functional studies to identify the full pathways of the triterpenoid synthesis, the P450s, and the glycosyltransferases to generate triterpenoids in cucumber. And so this has been done here. You can see some of the enzyme assays here, testing different lines of knockouts and look at how much triterpenoid is being formed. They looked at matching this to certain QTLs between different red cucumber lines. And they also looked at the SNPs that are actually determining these knockouts. And so this is of great interest because the bitterness has really been bred out over time out of the cucumbers. And so there's a great interest of understanding what are the target genes here, what are the target enzymes that are providing bitterness, to do this either in a more targeted fashion or to actually bring back some of the bitterness for the consumer areas where that is actually desired. So it's a really interesting paper that came out recently, and I'm happy to provide all of those references for those of you who might be more interested and want to learn more about this. Now, last but not least, our carotenoids or tetraterpenoids. So we're arriving at 40 carbons. They're made quite a bit differently now. So we have to look at that. What is similar is to the triterpenoids is that we're now using a condensation not of individual 5-carbon units, but we're actually doing a condensation of 2 molecules of GGPP. So 2 molecules of the diterpenoid precursors, the C20 precursors. And so for the diterpenoids, this precursor being located in the plastids, carotenoid formation also is occurring in the plastids, at least to the extent of preparing the hydrocarbon scaffold. Right? The function modification, again, is happening to some degree at the ER, to a large degree. So again, very similar to what we had just discussed for the head-to-head condensation for the triterpenoids, we're using the exact same reaction mechanism, essentially using the phytoene synthase. That's actually the enzyme that is taking 2 units of GGPP and is generating 1 long 40-carbon precursor for the carotenoids through this head-to-head condensation. What we do from there, right, yet again, here is your first reaction here. It's a condensation catalyzed by the phytoene synthase. You have this reaction now. The next step is actually a number of desaturations and isomerizations. What that allows you to do actually is that you are generating now a linear molecule with almost all of the double bonds being conjugated. Right? And so this is really important because what this conjugated double bond arrangement helps to do is it actually allows for the function of carotenoids to kick in. It will generate color, right? All your like yellow to red ranges of the different structures of carotenoids. So these desaturations, isomerizations toward a conjugated state are really critical for the coloring and the physiological functions of carotenoids. So it's a very critical step that is catalyzed here by several enzymes. Once you have arrived here at lycopene, at this intermediate here, you will do cyclization, sort of coming in at the ends here, using somewhat similar mechanisms for the cyclization as we had seen for the other terpenoids. So there are different kinds of these lycopene cyclases. What you will generate in the end is carotene, and you have either one cycle, but mostly you have carotene here as a major intermediate, having 2 6 cycles here, 2 6-membered rings at the tail ends of the lycopene molecule. And once you have achieved that, you're starting to actually introduce oxygen. You're doing a bunch of oxygenations. So a lot of this here will be catalyzed by P450s. There can be some more desaturation steps. For the most part, you're now starting to hydroxylate, especially the 6-membered rings here at the end of the molecule, generating, again, changes in the color, but also changing in the bioactivity of these compounds, moving all the way down to these different carotenoids with different functions. And so the general function of these is part in general metabolism, right? It's your electron transfer and photosynthesis. Carotenoids make up for the deficiency in chlorophyll in absorbing electrons in the largely greener spectra. So that's part of that, but also their capacity to transfer electrons is lying in these conjugated double bond arrangements. And so this also provides carotenoids with the activity of protecting against oxidative stress, specifically by scavenging radicals and reactive oxygen species. So that's a large part of their function. Damage that is created through UV irradiation, for example, carotenoids play a major role in preventing UV damage. So essentially, you can see them as a sunscreen in the plant. But also, right, attraction of pollinators and seed dispersers. So the pigment function of the yellow to orange-red colors of the carotenoids comes into play here in attracting pollinators and seed dispersers. Just to end on, like I showed you in the pathway there, you have basically two subgroups. You have the carotenes, these are the more unsaturated hydrocarbons. And then you have the xanthophylls and the carotenoids. These are your oxygenated compounds, and they are forming the natural pigments, again, ranging from yellow all the way to almost red. I'm just sort of showing some of the examples here, like lycopene and astaxanthin. Your red pigments, so this is what you would, for example, see in tomato. Alpha and beta carotene, these are really the bright orange colors that you would find in carrot, for example, in orange peppers, pumpkin, and so forth. And then the more yellow ones, lutein and zeaxanthin, these are the ones you would find in maize. I mean, you find them in all of these crops, but predominantly in maize, that gives you the yellow color. And just sort of as a fun fact, carotenoids also are, to a large degree, what makes your salmon red, what makes flamingos red. But here it's not really internal pathways. This is more what they're getting through their diet, eating organisms that actually are producing carotenoids. And so this is the end of this lecture. I just wanted to give you sort of a bit of, in a nutshell, some ideas on the commonalities and differences between these terpenoid subgroups, their differences in biosynthesis, function, and structure, and just to give some highlights and some examples. Their diversity is really too large to really cover all the aspects here.